Hey everyone, welcome to chapter 7. In this chapter, we're going to talk about genetics. We'll start with what is DNA. We'll talk about the structure of DNA, how we found the structure of DNA, and then we'll talk about how DNA codes for proteins, how we replicate DNA, cells replicate DNA, and how proteins are produced from transcription and translation of the DNA. So in the 1860s, Gregor Mendel, who is known as the father of genetics, was investigating pea plants and trying to figure out how certain characteristics were inherited. He concluded that plants transmit distinct factors to their offspring. He didn't know what those factors were, but today we know that those factors are genes. So what are genes? Genes are proteins that are encoded for in our DNA. So genes code for protein synthesis. Enzymes are proteins, and so genes code for enzymes. In 1941, George Beadle and Edward Tatum were two scientists that reported that genes direct the production of enzymes, and they did this by studying the mold Neurospora. Neurospora species can grow on minimal nutrients, and so they used Neurospora for that reason. They X-radiated the Neurospora to produce mutants that wouldn't be able to grow as effectively on minimal media. Then they grew the Neurospora on a nutrient-rich medium, which grew both the parents and the mutant progeny. They then took individual colonies and grew them on media that contained all the nutrients they may need minus one specific enzyme. And eventually they were able to identify mutants that had a specific metabolic defect where they couldn't produce one type of nutrient or enzyme, and so they couldn't grow. This allowed them to conclude that a single gene determines the production of a single enzyme, and they won the Nobel Prize in 1958 for this. So if you look at DNA, it is composed of 4 different nucleotides, so 4 basic letters: adenine A, thymine T, cytosine C, and guanine G. These 4 nucleotides code for a huge diversity in life. All living things contain the same DNA. So bacteria, all the way up to humans, have the same DNA. And we use the same amino acids as well. So there are 3 nucleotides that encode for 1 amino acid. And there are 61 different codes that code for amino acids. We have 20 different amino acids that make up every protein on Earth. So yeah, we're all very closely related. Bacteria all the way up to humans. We have the same genes, or same genetics. I won't say same genes. So what is the genome then? It is the complete set of genetic information. So this is the chromosomes within our cells. In animals, so humans, we are diploid at most chromosomes. Which means we have 2 of the same chromosome. One came from mom, one came from dad. And then we have, depending on the sex chromosome, if you are XX, you are diploid, but if you are XY, then you are haploid for the X chromosome, haploid for the Y chromosome. All cells have to contain their DNA because the DNA is what tells the cell how to produce new cells and tells the cell to make whatever proteins they need. In certain cells, like bacteria, they may have extracellular DNA known as plasmids. And so that would be also part of their DNA. So viruses can have either DNA or RNA, but they never have both. So viruses are not living though. So I said that all living things have the same DNA. Viruses aren't living, so I didn't put them in the group. The study of nucleotide sequencing is known as genomics. And this is a huge field because we're trying to identify every gene for every single organism on Earth. Right? So all cells need to be able to replicate to survive. And there are two basic functions that cells have to do so that they can survive and replicate. The first is they have to copy their DNA so that each new daughter cell will have a copy of the DNA. And the second is they have to be able to produce proteins. And protein synthesis is done through a process known as transcription, which is right here, and translation here, okay? So the central dogma of biology is that DNA carries the information for all of our genes, and DNA codes for proteins to be produced. So DNA codes for a protein, RNA carries out the information of the protein and actually produces the protein. So what does DNA look like? I'm going to do this really quick and then I'll stop this video. DNA is double-stranded. It's in a helical form, so it's wound up. The strands are anti-parallel to each other. So if you ever look at DNA, you can see here, you have a 5' phosphate on one end and a 3' hydroxyl unit on the other end. And on the opposite strand, you have the 3' hydroxyl unit that is attached to that 5' end, and a 5' or a phosphate attached to the 3' hydroxyl unit. So they run anti-parallel. You can see the sugars and phosphates are upside down on this one and right side up on this one. This is how DNA always runs. They're bound together, complementary. So we have a sugar known as deoxyribose that is bound to a phosphate. And then to the other side of the sugar, to a carbohydrate group, we have the nucleotides. And so you can be an adenine, a guanine, a cytosine, or a thymine. Adenine always binds to thymine. They are complementary. Guanine always binds to cytosine. They are complementary. They are bound by either 2 or 3 hydrogen bonds. And so when I say the term complementary, what I mean is that they have ends that fit together. So this could be an adenine, thymine, guanine, cytosine. So they fit together. Adenine and guanine couldn't fit together. Adenine and cytosine or guanine can't fit together, and thymine and guanine and cytosine can't fit together. So they just don't work that way. I mentioned that they're complementary to each other and they run anti-parallel, so they run in the opposite direction. One is upside down to the other one. I also mentioned that adenine and thymine bind together and cytosine and guanine bind together. So there's the very large picture of what I was showing you. I'm going to stop here because this has been almost 10 minutes and we'll get into our next video. Okay? Have a great day. Bye.